In this experiment, we will be determining the identities and concentrations of 5 unknown protein samples. In order to accomplish this, we will be casting our own SDS page gels for electrophoresis and then running our unknown proteins alongside a protein standard. Gel electrophoresis is a technique that is used to analyze and separate DNA, RNA, and proteins. The gel is loaded with the DNA, RNA, or protein samples, and an electric field is applied to the gel. This causes the molecules in the sample to move towards the electrode of opposite charge. Smaller molecules will move more quickly than larger, heavier molecules in the sample. Therefore, the smaller molecules will travel farther across the gel. The Software Image Lab has several features that can assist in analyzing the gels after imaging. It can be used to determine the molecular weight, relative front, and intensity of the bands in the gels. The first step of this experiment is to hand cast the polyacrylamide gels. The resolving solution is first made without APS or TMED. The apparatus for creating the gels is assembled and a mark is made about a centimeter below the comb. After adding the APS and TMED, the resolving solution is transferred to the apparatus using a pipette. It is filled up to the marked line and isopropanol is immediately added on top. The resolving gel is allowed to polymerize for 5 minutes before the remaining alcohol is removed using filter paper. Meanwhile, the stacker solution is prepared. It is poured on top of the resolving gel and the comb is carefully inserted. The stacking gel is allowed to polymerize for 30 to 45 minutes. Before running the gels, the given proteins A, C, D, G, and J must first be prepared. Lamely buffer and beta-mercaptoethanol is added to each protein sample stored in microcentrifuge tubes. The proteins are then heated at 95 degrees Celsius for 5 minutes in order to denature the proteins. Once the stacker polymerization is complete, the combs are carefully removed from the stacking gels and the gels are loaded into the electrophoresis cell. The outer chamber is first filled with SDS buffer up until the fill line. The protein samples are then loaded into the wells using a micropipette. At least one of the wells in each gel is loaded with a protein standard. The inner chamber is then filled up to the top with buffer before the cell is connected to a power supply. The gel is run at 150 volts until the proteins reach the resolving gel. The remainder of the run is done at 300 volts. When the front has nearly reached the bottom of the gel, the power supply is turned off and disconnected. The gels are removed from their glass cassettes and transferred onto a tray for imaging. The resulting images are then analyzed in Image Lab. The gel images are first given a false color to make the bands more visible. Then the lanes and bands are all labeled using the appropriate Image Lab tools, taking special care to correctly label the standards. Then, the analysis of the data can begin using Image Lab software. Image Lab creates a table which gives an estimation of molecular weight. The average values were found for the unknowns except for protein D, whose band did not appear in any of our trials. The software can also give values of volume or intensity. Together with a calibration curve of these intensities versus known protein sample masses, which can be seen here, concentrations can be found by finding relative mass and then dividing by the known sample volume, which was 15 microliters in our case. The first trial gave a large amount of bleeding in C. Therefore, we made dilutions of it in order to determine the best concentration for imaging C. We made dilutions by simply increasing the ratio of Lamely buffer and beta-mercaptoethanol to the C that we were given. The image shown here is the last trial for which we chose to do our analysis on, and this includes the proper dilutions of C. Then, purities can be found by summing the intensities of the bands, not including the last, and dividing by the total intensity in each lane. Only the 50 times dilution of protein C was used to determine its purity. Protein D is omitted due to lack of data. We suspect that protein A was casein, including beta-casein and alpha-casein, because there were 2 distinct bands in A every time, very close to each other. We suspect that protein C was BSA. We suspect that protein D was myoglobin. Even though we did not see it, we suspect that because it had such a low molecular weight, it ran off of the gel every single time, which means that it would have to be myoglobin. We think that G and J were actually the same protein, ovalbumin, due to their very similar molecular weights. 0.34% for A, 35.34% for C, 57.31% for G, and 29.35% for J.